Today we discussed about plus 2 biology, chapter 4, principles of inheritance and variation. This lesson deals with the term heredity, variation, Mendelian laws, its deviations, chromosomal disorders, etc. If you like this class, please like, share and subscribe. Then we start our lesson. We can see lots of similarities and differences between parents from offsprings. Offsprings means individuals or children. Children of same parents and between the members of the population, these similarities and differences are seen. Then what is heredity and what is variation? Heredity, the transmission of characters from one generation to the next, that is from parents to offsprings, is known as heredity. Characters means all type of characters, physical characters, behavioral characters, intelligence, beauty, looks, etc. are termed characters in genetics. The transmission of characters from one generation to the next, that is from parents to offspring, is known as heredity. Then what is variation? Variation means difference, that between parents and their offsprings or between offsprings of the same parents or between members of the same population. Variation means difference between parents of their offsprings or between offsprings of the same parents or between members of the same population. Then first we should need to know some terms that usually used in genetics. There are some technical terms in genetics. First term, factor. Factor. Factor means inheritable units, that is gene. Factor means inheritable unit. That means genes. What is gene? Genes are located on DNA. Genes are the pieces of DNA DNA is the part of chromosomes. Chromosomes usually seen in nucleus. Factor means inheritable unit. That means genes. Second term, gene. What is gene? A segment of DNA molecule which determines the unit of inheritance. A segment of DNA molecule which determines the unit of inheritance. Next term, alleles or allelomorphs. Alleles or allelomorphs. Two or more alternative forms of a gene are called alleles. Two or more alternative forms of a gene are called alleles. Alleles. Next term, trait. Trait is the morphologically or physiologically visible character. It is the morphologically or physiologically visible character. Example, flowers color. Flowers color, skin tone, length of hair, etc. etc. are considered as trait. Then factor is the inheritable unit, that means genes. Genes are the segments of DNA which determines the unit of inheritance. Alleles or allelomorph, two or more alternative forms of genes are called alleles. Example, if we take the height of the plant, there are two or one pair of genes responsible for the height. So, we can write this. Trait is the morphologically or physiologically visible character. What is morphology? It is the outward visible form, that is height, skin tone, skin color, etc. Physiology means internal activities, that is digestion is a physiological activity. Next, dominant trait. Dominant trait. Out of the two alleles or allelomorph of the trait, the one which expresses itself in heterozygous organism, that is F1 hybrid, is called dominant trait. in case of heterozygous condition. Example, this is denote the heterozygous condition of height of the plant, tallness of the plant. Here one dominant and one recessive gene that represent the height of the plant. But this plant, the morphology of the plant is tall, that the outward feature is tallness. In case of heterozygous condition, in F1, the expressive character is dominant trait. Dominant trait in case of heterozygous condition. This is an example of heterozygous condition. The one gene expresses itself is called dominant trait. Here capital T is the expressive character. That is, this type of plants are always tall. Then recessive trait in heterozygous condition. The allele with two type of genes. In heterozygous condition, one gene suppressed by the activity of another gene. Here example, capital T, small t is recessive. This represents the dampness. This dampness is suppressed by the activity of this capital T. So this plant is always tall. Next term, genotype, that is the genetic constitution of an organism. This only examined under microscope. In this heterozygous case, we can see all plants, heterozygous plants are tall, but we check this gene or allele under the microscope, we can understand this allele with two types of genes. Next term, phenotype, the outward appearance of a trait, that is the height of the flower color, etc. The outward appearance of the trait or character. Next, homozygous condition, an individual possessing identical alleles. This is homozygous condition of tallness, an individual possessing identical alleles. Heterozygous condition, in individual with dissimilar alleles. Next term, parental generation, parents used for first crossing. Example, we can cross a tall plant with a dwarf plant. These tall and dwarf plants are considered as parent generation, P1. F1 generation, that is first filial generation, the progeny produced from cross between two parents, the first generation. F2 generation, the progeny is produced by the selfing of F1. We can discuss monohybrid and dihybrid cross, then we get more explanation. Monohybrid cross, considering for a single pair of contrasting character for a cross. That is, in monohybrid cross, there are 17 contrasting characters in pea plants, but for monohybrid cross we only consider one contrasting character, that is height, flower color, position of flower, etc. etc. Next term, dihybrid cross, considering two pair of contrasting characters for a cross. In dihybrid cross, we can choose height of the plant and flower color. That is two contrasting characters. If we take two contrasting characters for a test, such tests are known as dihybrid cross. Next term, hybridization, that is artificial crossing. Test cross. Crossing of F1 with recessive parent. Crossing of F1 with recessive parent. Tall plant, dwarf plant, gametes, then we get F1 generation, that is heterozygous tall. We cross this F1 with recessive parent. This is dominant and this is recessive parent. We cross F1 with recessive parent. Such cross is known as test cross. We get one tall and one dwarf. This test is usually used for checking the purity of the gametes. We should take homozygous parental generation for testing or crossing. Next, reciprocal cross. We know the pea plants are bisexual flowers. Pea plants with bisexual flowers, that is flower with male and female reproductive parts are seen in same flower. Such flowers are known as bisexual flower. So we can take any plant as male and female. In first test we take this plant as female, then next test we can take it as male plant, male or female, that is we can alternate, but the result is same. Reciprocal cross means crossing in which the sex of the parents reversed. The result is same. Next, the 7 contrasting characters used for the test. 7 contrasting characters. Here is the 7 contrasting traits or characters used by Mendel. These are the 7 contrasting characters and these are the alternatives. First contrasting character, stem height. It may tall or dwarf. Flower color may violet or white. Flower position, axillary flowers or terminal flowers. Pod shape, inflated or constricted. Pod color, green or yellow. Seed shape, round or wrinkled. Seed color, yellow or green. These are the dominating characters. Tall, violet, axillary flower case, inflated, pod shape, green, pod color, round seedness, yellow color of the seed are the dominating characters. These are the recessive characters. We use the first letter of the dominating characters for test. Next topic, mental success. There are so many scientists conduct experiments in genetics, but Mendel succeeded. Why? Because he chose pea plant as the study material, Pisum sativa, pea plant. The pea plants are cosmopolitan in distribution because they are seen all over the world. Then they have 7 contrasting characters. They are annuals. They complete their life cycle within 6 months or 1 season. They are annuals. They complete their life cycle within 1 season. They are bisexual flowers. So it helps for selfing. Pea plants are bisexual. The flower with male and female reproductive organs are seen. So it helps for selfing. Next, Mendel's methodology. The steps of crossing, that is Mendel's methodology. First, homozygous pure-line parents with contrasting characters were crossed. Homozygous pure-line parents are selected. Homozygous pure-line parents are selected. That is for height, by conducting test cross we can determine the purity of parents. Next, self-pollination was prevented by removing the stamen of the female flower. Self-pollination was prevented by removing stamens of the female flower. That is known as emasculation. Emasculation of female flower. The pollen grains are removed at the stage of bud. Third point, after pollination, that is artificial pollination, desired pollen grains are dusted on the stigma of the female flower. That is known as artificial pollination or hybridization. After hybridization, female flowers are bagging separately. After pollination, All female flowers are bagged by polythene sheet or butter paper. Then seeds were collected as F1 generation. Seeds are collected, then selfing of F1 to get F2 generation. These are the steps conducted by Mendel. That is known as Mendel's methodology. Then we can discuss about one monohybrid cross. Monohybrid cross. We can choose flower color. The trait flower color. Take violet flower and white flower. This is the dominant, this is the recessive parent. This is the dominant parent and this is recessive parent. So these are known as parental generation. We take the first letter of the dominant character. All characters depend upon one pair of allele. Then we get, this is considered as female and this is male. These are gametes. In case of male, pollen grain is a gamete, haploid gamete. Here egg cell is the haploid gamete. Then we get F1 generation, that is usually violet. This is heterozygous condition. Then next step, selfing of F1. That is selfing of F1. Capital V, capital V, capital V, small v, capital V, small v, and small v, small v. Here this is homozygous violet, 2 heterozygous violet and 1 white. Then we write the phenotypic ratio. That is 1 homozygous violet and 2 heterozygous violet, but the appearance, all 3 of them are violet color. So phenotypic ratio 3 violet and 1 white. Then the genetic constitution, that is genotypic ratio, we get 1 homozygous violet, 2 heterozygous violet and 1 white. So in case of monohybrid cross, we get phenotypically 3:1 ratio and genotypic ratio 1:2:1. Again, another monohybrid cross that we consider the character tallness. This is homozygous tall plant, maybe male or female. Then this is homozygous recessive parent, that means dwarf male female. This is the parental generation, and in case of female plant, pollen grains are the gametes haploid. Egg cell is the gamete of female case, that is egg cell. These are known as gametes. Then we get F1 generation, that is heterozygous tall. Heterozygous tall. This is homozygous tall. This is homozygous dwarf. This is homozygous dominant, homozygous recessive. Then selfing of F1. F1, that means one homozygous tall plant, two heterozygous tall plant, and one dwarf plant. So phenotypic ratio 3 tall and 1 dwarf. Genotypic ratio 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. Then 3 laws that proposed by Mendel. They are law of dominance, law of segregation, and law of independent assortment. We can discuss about 3 laws. First law, law of dominance. Law of dominance. When the two genes of a pair represent a contrasting character, the expression of one is dominant over that of the other. When two genes of a pair represent contrasting characters, the expression of one is dominant over the other. In case of heterozygous condition, this is heterozygous condition. In case of heterozygous condition, the two genes of a pair represent contrasting characters, that is tallness and dwarfness. Representing contrasting characters. Here the contrasting character tallness and dwarfness. in case of heterozygous condition, example tallness, the two genes of a pair represent contrasting characters, tallness and dampness. The expression of one dominant over the other. The expression of one dominate over the other. So in this case the plant is tall, dominate over the other gene. This law is known as law of dominance. In case of heterozygous condition, the two genes of a pair represent contrasting characters, that is tallness and dampness. The expression of one gene is dominating over the other gene. That is known as law of dominance. Next law, law of segregation or purity of gamete. This law is known as universal law. Second law, law of segregation or purity of gametes. This law is also known as universal law. During gamete formation, in all sexually reproducing organisms, plants or in case of animals, the two factors or genes of a pair alleles passing into different gametes. If we take this case, during gamete formation, in all sexually reproducing organisms, the two factors or genes of an allele passing into different gametes. This is known as law of segregation or purity of gametes. During gamete formation, in all sexually reproducing organisms, the two factors, that is genes, genes of a pair, that is alleles. This is the allele or pair of genes that represent the character tallness, alleles passing into different gametes. Passing into different gametes. This is diploid case. Gametes are haploid. This may pollen grains or may eggs. During gamete formation, in all sexually reproducing organisms, the two factors or genes of a pair or allele passing into different gametes or two gametes. Next topic, dihybrid cross. If we take two contrasting characters for a test, such tests are known as dihybrid cross. In our textbook, this is the picture of dihybrid cross. Here take round yellow seeded plant crossed with wrinkled green seeded plant. Dihybrid cross. Here we take a round yellow seeded plant. The seed is round, the shape of the seed is round and color yellow, crossed with wrinkled seed, green colored wrinkled seed plant. Green wrinkled seeded plant. Round yellow seeded plant crossed with wrinkled green seeded plant. Round yellow seeded plant crossed with wrinkled green seeded plant. These are the parental generation. This is the dominant character, this is the recessive character. First letter of the dominant character are taken. So capital, round, capital R, allele, that represent the roundness, and yellow color of the seed, that also the seed color also controlled by a pair of genes. Then here in recessive case we take the small letter of this dominant character, that is wrinkled smaller and green small y. This is the dominant character. Round and yellowness are the dominant characters. Wrinkledness and green color are the recessive character. So always we take the first letter of the dominating character, that is round, capital R, that is a pair of genes are controlling the round shape of the seed, and yellow color of the seed is controlled by one pair of gene or allele, that is capital Y. Then take the small letter of recessive characters. We can take, it may male or female, or we can reciprocate. We can take this is male and this is female. No problem, because this is a bisexual flower. Then we get the gamete, half number of chromosome. Gametes with half number of chromosome, that is in case of female this is the X, and here small r small y, this is the pollen grains, gametes. Then we get F1 generation, that is capital R, capital Y, small y. This is heterozygous round yellow seeded plant. All capital letters are dominating. And these are the dominating characters and these are the recessive characters. All dominating characters usually suppress the activity of the recessive characters. So in this heterozygous condition we get round yellow seeded plant. Yellow seeded plants as F1. All of them are heterozygous. So here two types of genes represent each and every character. So it is heterozygous. Next step, selfing of the F1. Selfing of the F1 is the next step. Selfing of the F1 is the next step. That is capital R, small r, capital Y, small y into capital R, capital Y, small y. Selfing means this is also bisexual flower. The pollen grains of this plant is dusted on the stigma of the same flower. That is selfing. Here we get it for gametes. That is capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, capital R, capital Y, capital R, small y, small r, capital Y, small y, small r. These are gametes. 4 types of gametes are expected. Again, here also capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. We get 16 types of progenies in F2 generation. So we should use a checkerboard or Punnett square. We should write or draw a Punnett square with 16 columns. Here we take the female gametes and here male gametes in this row, or vice versa. Just we put capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Then here these are the excels and these are the pollen grains. Capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Then we cross this gamete with all these rows. Then capital R, capital R, capital Y, capital Y. Then capital R, capital R, capital Y, small y. Then capital R, small r, capital Y, capital Y. Next capital R, small r, capital Y, small y. Next row capital R, capital R, capital Y, small y, capital R, capital R, small y, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y. Next capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, small r, capital Y, capital Y, small r, capital Y, small y. Next capital R, small r, capital Y, small y, capital R, small y, small y, next small r small r, capital Y small y, last small r small y small y. Here round yellow, all dominant character represent the capital letter. All capital letter represent the character. That is the expressing character. Round yellow, round yellow, round yellow, round yellow. All are round yellows. Then round yellow, round green, because all these two genes are recessive, so it is green. That is round green. This is round yellow. Here also round green. Next round yellow, round yellow. Here wrinkled. Two recessive genes represent the wrinkledness. Wrinkled yellow. Here wrinkled yellow. Round yellow, round green. This is wrinkled yellow. This is wrinkled green. Now we check the phenotypic and genotypic ratio of dihybrid cross. Phenotypic ratio of dihybrid cross. That is, we get all homozygous and heterozygous case, we get 9 yellow round seeded plants. Then 3 wrinkled yellow seeded plants. Then 3 green yellow seeded plants and only 1 wrinkled green seeded plant. That is the phenotypic ratio. 9 dominating character round yellow seeded plant, 3 wrinkled yellow seeded plants, then 3 green yellow seeded plants and 1 green wrinkled seeded plant. This is the phenotypic ratio. Then next, genotypic ratio. If we examine the genetic constitution of all these 16 plants, we get another different ratio. That is 1:2:1, 2:4:2, 1:2:1. Altogether 16. This is the genotypic ratio and this is the phenotypic ratio. This is the dihybrid cross. We can take any different types of cross: tallness, flower color, flower position, and pod color, pod shape, seed shape. There are 7 contrasting characters. We can take any 2 contrasting characters for dihybrid cross. Here we take color and shape of seed. Next, law of independent assortment. Third law. Law of independent assortment. Law of independent assortment. In this law, if we consider 2 pairs of contrasting characters, that is considering the dihybrid cross. If consider 2 pairs of contrasting characters, that is tallness or flower color. Here we take the seed color and seed shape. Any 2 contrasting character. Genes for each character passing to next generation without any type of mixing. That is, the genes for tallness never mix with the genes for flower color. If consider 2 pairs of contrasting characters example, tallness and flower color, tallness of plant and flower color, genes for any type mixing, genes for any type of mixing, that is genes for tallness never mix with the genes for flower color. Consider 2 pairs of contrasting characters for crossing. Genes for each characters, genes for each characters passing to next generation, passing to next generation without any type of mixing, without any type of mixing, that is the genes for tallness never mix with genes for flower color. Again, if consider 2 pairs of contrasting characters, example, take tallness and flower color for crossing, that is dihybrid cross. Genes for each character passing to next generation, that means to F1 or F2 generation without any type of mixing. That is, genes for tallness never mix with genes for flower color. That is the law of independent assortment. Next topic, chromosomal theory of inheritance. Chromosomal theory of inheritance. What is chromosomal theory of inheritance? There are certain points we should consider. This theory introduced by T.H. Morgan and his colleagues. Morgan worked with Drosophila melanogaster, that is fruit fly, because they could be grown on. They take Drosophila as study material because this Drosophila could be grown on simple synthetic medium in laboratory, easily grown. They complete their life cycle in about 2 weeks. Single mating produce large number of progenies or children. Male and female flies are easily distinguishable. It has many types of hereditary variations can be seen with low power microscope. This theory introduced by T.H. Morgan. Chromosomal theory of inheritance. This theory introduced by T.H. Morgan and his colleagues. They take Drosophila melanogaster or fruit fly as study material. Because Drosophila could be grown on simple synthetic medium in laboratory. They can grown on simple medium in laboratory. Second, single mating produce large number of progenies or children. Single mating produce large number of progenies or young ones. Third point, they complete their life cycle within 2 weeks. Life cycle complete within 2 weeks. Male and female flies are easily distinguishable. Male and females are easily identified or distinguishable. It has many types of hereditary variations. it has many types of hereditary variations. That can be seen with a low power microscope. Then what is chromosomal theory of inheritance? The somatic cell, that is body cell of an organism, which are derived by repeated mitotic division, that is normal division or zygote, have two identical set of chromosomes. They are diploid. These chromosomes derived from parents, that is maternal and paternal. Humans have 23 pair of chromosomes. That is the first point. Chromosomal theory. First point. The somatic cell, that is the body cell, not the reproductive cell. Reproductive cells are formed in human case, that is testis and ovary, but other parts are we considered as somatic body or somatic cells. The somatic cell or body cell of an organism, any organism, which are derived by repeated mitotic division, that is usual division, one cell divided into two, that is the mitotic division that occur in somatic cells. Division that occur in zygote have two identical set of chromosomes. They are diploid. In case human case, 23 pair of chromosomes are seen in all somatic cells, that is 2n, diploid. But in gametes, this is only 23 number, not pair. The chromosomes derived from parents. These two set of chromosomes, one set is derived from father, that is paternal, and the other is from maternal, that is from mother. The somatic cell that is also known as body cell of an organism which are derived by repeated mitotic division of zygote. Mitotic division of zygote. What is zygote? Zygote is the product of fusion of two haploid gametes, egg and sperm. In plants, egg cell and pollen grains. Gametes are haploid. Two haploid gametes fused to form first diploid zygotic cell. Have two identical sets of chromosome. They are diploid. All somatic cells are diploid. That is 2n. In human case, it is 23 pair. These chromosomes derived from, these paired chromosomes derived from parents. that is maternal and paternal. Half from maternal from mother and half from paternal or from parent, father. Maternal and paternal. 23 from father and 23 from mother in case of humans. That is 2n case. Humans have 23 pair of chromosomes. Next point about chromosome theory. The chromosomes of homologous pair separate out during meiosis at the time of gamete formation. The chromosomes of homologous pair separate out during meiosis, that is reduction division that undergoes in reproductive part at the time of gamete formation. Second point, the chromosomes of homologous pair separates this is the homologous pair, one from father and one from mother. This is one homologous pair. Consider this is one of the homologous pair. At the time of gamete formation, half of them enter into the gamete. Homologous pair of chromosomes with similar type of genes. One from mother and one from father. This is the diploid case. At the time of gamete formation, meiosis or reduction took place. Half number or half of this homologous pair detached from each other and enter into gamete. Next point, genes are located linearly on the chromosome. Chromosome is made up of molecules of DNA and a specific set of segments of DNA are genes. Third point of chromosome theory. Genes are located linearly on the chromosome. Chromosome is made up of molecules of DNA.